So a point mutation is when a single base pair is altered or mutated. In this video, I'm going to talk about 4 types. There's missense, nonsense, silent, and read-through. To the right, there's a diagram that helps me remember the transition and transversion phases. So you have the pyrimidines, which are C and T, and the purines are A and G. So a transition from a purine for a purine is A to G or G to A, and then pyrimidine for pyrimidine is C to T or T to C, and transversion is purine for pyrimidine and vice versa. The top sequence is the DNA sequence of the wild type that was given to us for this prompt. I then translated to mRNA by changing the T's to U's, and then I grouped them into 3, looked on the amino acid table, and found the corresponding amino acid. For comparison purposes, I'm going to keep the wild type DNA that was on the previous slide at the top. So for this nonsense mutation, a nonsense mutation is a change of a normal amino acid codon for a stop codon. So I changed the C to an A in cytosine in the wild type and then translated it to mRNA, which gave me UGA, which codes for the stop codon. So we have a stop codon where the cytosine used to be. So since I changed it from a C to an A, it's going from a pyrimidine to a purine, causing transversion. Next we have silent mutation, and this does not change the corresponding amino acid or function of the proteins. So you're going to change the base pair of the wild type, which I changed it from a C to a T, translated that into mRNA, so I got UGU, which also codes for cytosine. Since I changed it from a C to a T, I'm changing it to a pyrimidine, which is transition. So this slide is showing missense mutation. A missense is a point mutation causing the change of one base pair that results in a different amino acid. So as you can see, the top cytosine, the T, I changed that to an A down in my mutation. So that translated that A to mRNA, causing it to be A again. So AGC on the table translated to serine, so it went from cytosine to serine, so a T to an A, which is transversion because it's going from a pyrimidine to a purine. Finally, we have read-through mutation. So read-through mutation is a standard stop codon that is converted into amino acid. So the stop codon in the wild type, it got changed from a T to a G in the DNA. So I translated that into the mRNA, which I still got G. So I got GGA, which changed the stop codon to glycine. So then I continued the chain and I got proline and glycine again. So my mutation was from a T to a G, causing a transversion.